Welcome, students, to the last part of Chapter 3, which will focus mostly on the nucleus and kind of what goes on in the nucleus. Part 2 of this chapter, Chapter 3, focuses on the cytoplasm. And essentially what we're talking about with the cytoplasm is pretty much everything else that's not the plasma membrane and not the nucleus. So it's kind of everything in between that includes the cytosol, which is kind of the fluid gel that is in all of our cells, and everything kind of dissolved in that. Proteins, salts, sugars, any other type of electrolytes, anything that's water soluble is going to dissolve in the cytosol and be in the intracellular fluid. The cytoplasm also includes what we call inclusions, and these are things that are insoluble in the cytosol, and kind of which cells have which inclusions is different. Not every cell has the same inclusions. Things like pigments or fat droplets or kind of maybe water vacuoles, any kind of maybe say calcium crystals or little granules of glycogen. There's all kinds of different types of inclusions, and kind of how many and of what type depends largely on what type of cell we're talking about. And then lastly, which is the biggest part of this part 2, are all of the cellular organelles. And these are all of the kind of metabolic machinery of the cell. All of the things inside the cell that makes the cell function. They all have a very specialized function. Some of them are membranous, meaning they're somewhat contiguous with either the nuclear envelope or the plasma membrane. Some of them are non-membranous, and there are a ton of them, and there's a really lovely table in your book that lists all of the different organelles: the mitochondria, the ribosomes, the endoplasmic reticulum, the Golgi, the peroxisomes, the lysosomes, all of the different cytoskeletal elements, and the different types of cellular extensions like cilia and flagella. And sure, I could go over this with you guys, and I can sit here and say the mitochondria are these double membrane structures that have these little folds called cristae, and they are the site where ATP is made. And sure, yeah, I could do that, but I think you guys are a whole lot smarter than that. And so I will leave you on your own to go over the different cellular structures. If you've ever had a biology class, you've gone over these things before, and this nice little table here gives you not only a little bit of kind of what they look like, including a little diagram, but also what they do. And that is generally what I will ask you guys, is can you tell me what each of these things do? Give me a brief little blurb about kind of what they are. And yes, we could go over that. And certainly if you have questions, you can email me or ask in class when I see you guys again. But I think you all are plenty of smart enough to go over part 2 of chapter 3 on your own. So really what I'm going to focus on is part 3, because this contains some stuff that I think is very confusing to students, but that is also very important. And part of the reason nuclei are so important is this is like the brain of the cell, right? If you think of the brain as the boss of your body, well, the nucleus is the brain of each of your individual cells. This is where all your genetic information is. This is what makes you you. And so it's really quite important. You'll see it is encased in a little kind of what we call nuclear envelope, and your DNA, when your cells are in a certain part of their cell cycle, exists as what we call chromatin, which is essentially all of these kind of fibers that exist inside the DNA, and it's all nucleic acids. It's all DNA, just in a very kind of fibrous format, not well packaged, or at least not very tightly packaged. Now, in other parts of the cell cycle, it will get nice and neatly, tightly packaged into what we call chromosomes, and I'll show you what that looks like in just a little bit. But nuclei are really incredibly important because they do have the DNA. They do have all of your genetic information. So most cells have one nucleus, so they're uninucleate, and there are a couple in our body that aren't, but usually animal cells just have one nucleus. Some cells are multinucleate, skeletal muscle cells when they are formed. A bunch of cells merge together to make one skeletal muscle fiber, and so skeletal muscle cells end up with multiple nuclei. The same thing happens with some of the cells in our bones. They end up kind of merging together and become these like huge multinucleate kind of bone-eating cells, and then some of our liver cells kind of fall into that same package. And then there are some cells in the body that are anucleate, without a nucleus at all, like our red blood cells, for instance. Your red blood cells are basically just a sack of hemoglobin. They have lost all of their organelles, and they have lost their nuclei, although they don't start off that way. And you'll get a chance to kind of talk about red blood cell development in A&P 2. So this is what the cell cycle looks like, the life cycle of a cell. And you'll see it's really broken down into two main phases, what we call interphase and what we call the mitotic phase. Interphase is the cell basically doing its job. Whatever type of cell it is, this is when it is doing whatever it is that it's supposed to be doing in your body. It is broken down into a couple of different phases. There's a G1 or growth phase. There's an S phase. That S stands for synthesis, because this is where your DNA is replicated. And then there's a second kind of growth phase where the cell is kind of concerned with getting ready to go through and enter the mitotic phase. And the mitotic phase is actually two separate events. There's mitosis, which is the splitting of the nucleus, and then cytokinesis, which is the splitting of the cell itself. So if we look at DNA synthesis, this is roughly in a somewhat simplified version of what it looks like. In order for all of our bodies to grow and repair, our cells go through mitosis. We get and cytokinesis. We split we replicate our DNA, we split our nuclei, and we split our cells, and what started off as one parent cell is now two genetically identical daughter cells, and that is how our bodies are able to grow and repair. Well, part of that process is, of course, replicating the DNA such that each daughter cell has the identical DNA. So here you can see, here's our little chromosome, and you can see here's our double helix of our DNA. Because the DNA is kind of wrapped in this helical format, the first thing that has to happen is we have to unwind that helix. And so there's an enzyme that unwinds the helix, and that creates what we call a replication fork or a replication bubble. And then there is another enzyme, it's called DNA polymerase, that actually adds the new nucleotides using the old, like the quote-unquote old strands as a template to make the new strands by base pairing our nucleotides. A with T, G with C, C with G, T with A, etc., etc. So since we have two different strands of DNA. Right, we have what we call the leading strand, which is synthesized continuously. DNA polymerase basically just comes and chugs along, adding nucleotides until we have a completely new strand of DNA. And then we have what we call the lagging strand, which is synthesized in a kind of the opposite direction, right? See how it's going this direction? And the lagging strand is actually made in fragments. DNA polymerase comes along and adds nucleotides, and then another one will actually come along and add more nucleotides, and you end up with what we call Okazaki fragments, named after the man and wife that kind of discovered this process, or at least first described this process, and then a third enzyme comes along and kind of stitches it all together. But the result is that we have two new strands of DNA, but they contain kind of both an old strand and a new strand. So our two new helices have an old strand of DNA and a new strand of DNA. And so this is considered semi-conservative replication because half of the new strand is, or the new helix, is an old strand of DNA. Once we have our DNA replicated, the cell, still in interphase, can kind of do those final preparations for going to be about to split. We get our little centrosomes with our two little centrioles, which are part of our cytoskeletal elements. They're going to be very active at this point. During interphase, you'll see the DNA is in that chromatin form, kind of fibrous and fluffy. But then we enter the first stage of mitosis, and that is prophase. During prophase, the nuclear envelope will start to fragment and split apart. It's kind of what you see going on here. The chromatin will condense and kind of coil up into very discrete chromosomes, and those centromeres and the centrosomes will start to develop what we call the mitotic spindle, which is going to help pull apart the chromosomes. Each chromosome consists of what we call two sister chromatids, which are basically the kind of identical DNA from the replicated DNA. By the end of prophase, the chromosomes are attached to the mitotic spindle by what we call kinetochores. The nuclear envelope has essentially completely broken apart at this point, and the chromosomes kind of start to migrate to the middle of the cell. In the second phase of mitosis, it's called metaphase, all of the chromosomes are lined up on this imaginary line that we call the metaphase plate. The mitotic spindle is fully formed, and now we can actually finally start the separation process, which is what occurs in anaphase, the third phase of mitosis. Basically, these little kinetochore microtubules pull apart the two identical sister chromatids and essentially start to kind of walk them to either side of the elongating cell. You can see the cell is getting really, really, really long compared to what it was way back here. Once the sister chromatids are completely separated, as you see, then we have two new kind of daughter chromosomes identically going towards each end of the cell. That is when we enter the last phase of mitosis, which is telophase. That chromosomes have stopped moving. It's basically prophase kind of backwards. The chromosomes will uncoil back into chromatin. The mitotic spindle will disappear. We'll get new nuclear envelopes kind of forming around the kind of unraveling chromosomes. and cytokinesis will take place. Cytokinesis in our cells occurs by what I call a cleavage furrow, where essentially you just kind of pinch it off in the middle, and that separates the two new daughter cells, and we end up with two identical daughter cells when we started with one single parent cell. And that is mitosis. And pretty much every cell in your body, except the sex cells, the gametes, goes through mitosis in order to do growth and repair. The sex cells go through a slightly different process called meiosis, and so they end up different and not genetically identical. But in mitosis, one parent cell results in two genetic identical daughter cells. I'm going to actually stop here, and I'll put this last piece, which, if you know anything about the word central, think important. This eukaryotic central dogma is kind of vital to understanding not just human bodies, but pretty much all multicellular eukaryotic organisms. So let's put that in a separate lecture, so this one doesn't get too overly long and too overly huge in terms of file size. So we'll stop there for right now.